Hello students, welcome you all to a new video of Sunanda's tutorial. In this video we will discuss about micropropagation technique. So at first it is a technique of advanced plant tissue culture. It is a practice of rapidly multiplying plant material to produce a large number of progeny plants using new modern plant tissue culture techniques, and these plants are genetically identical. Okay, the micropropagation technique can produce many plants in a very short time, and those progeny plants are genetically identical. This technique involves cultivating small piece of plant tissue. So generally in micropropagation technique, we took a very small piece of plant tissue called as explant on sterile nutrient rich growth medium under controlled environmental condition. Then we grow that short plant tissue in a sterile nutrient rich growth medium and in controlled temperature, pH and humidity. Okay, next, so what are the stages of micropropagation? At first, initiation, we have to collect a plant, a healthy plant explant like shoot, tip, leaf or bud. It is carefully sterilized after collection and placed on a culture medium to start the process of multiplication. Next, multiplication or growing process. The explant is then transferred to the medium with specific ratio of hormone. So generally the auxin and cytokinin ratio plays important role because cytokinin is required for shoot growth and auxin is required for root growth, root development. So at first we have to place that explant in a cytokinin rich medium to create or to develop new shoot. Then we have to transport it into auxin rich medium to develop root. Okay, then acclimatization. Then after the development of shoot and root, a new small plantlet is developed. Then the newly formed plantlets are carefully transitioned or transported from sterile high humidity lab environment to the soil, that means into the environment. So these are the diagrammatic representation of different stages of micropropagation technique. After acclimatization, we can completely expose our plants to field condition. Now what are the key benefits of our micropropagation technique? So at first, mass production. It allows for rapid multiplication of plants in relatively small space and in a short time. It can produce disease free plants because we grow it in lab condition, in microbe free condition, and generally tissues like apical meristem is collected for this micropropagation technique. Then conservation, it is highly valuable for propagating rare and endangered plant species which are difficult to grow using seeds. Next, year round consistency. Because it occurs in a lab setting, production is not limited by seasonal changes. So we can grow it, we can cultivate it anytime. So these are some key benefits. And what are the limitations? While highly effective, micropropagation has a few drawbacks like high cost. So setting up a sterile tissue culture lab and equipments and this process require expertise. So it requires a high cost, high expense. Then contamination risk. The nutrient rich media can also easily support bacterial and fungal growth which can destroy the entire process. Then genetic variation. Under certain conditions, some culture method can lead to unintended mutations or variations in cloned plants. So these are some drawbacks or limitations. But you have to remember that this micropropagation technique completely rely on a capability or inherent capability of plant cell, that is totipotency. It is the inherent ability of the plant cell that a plant cell can divide and differentiate to form a completely new plant or functional plant body. Okay. So with this I am concluding my lecture video here on the topic micropropagation. If you liked it, please hit the like button and share with your other friends, and please subscribe to my channel Sunanda's tutorial. Thank you.